of this room on Friday. So they said that before this class on Friday, it's available, and after this class on Friday, it's available. So I had them block out essentially from 1:30 to 3:30. So my hope is some of you can get here early, some of you can stay late. And that way, if you can have that little bit of extra time just to breathe, I like to provide extra time. Can some people get here early? Can some people get here, stay late? So hopefully there's an option where you can do either or and not have it too disruptive. So my plan is to get here around 1:30 on Friday with the idea that you can start taking the exam. That means that when you do come into the room, you need to be very quiet, please. And just realize other people have already started, okay? So today's session right now, I'd like to spend most of it on thinking about the exam content and working through any questions you may have. Let's see, can I do that? Let me go through some of the things that I was finding on the first exam. So I haven't totally finished all the stats, but I did pick out the ones that were tripping up more people than not. And so textbook reading, I'm not terribly surprised. Textbook readings, I know that that's not, the main focus of this class. I don't make, so when some people buy textbooks, you can buy the Associated Test Bank with it. I just don't use those. And so those test banks really focus on the book stuff, but I make my exam from scratch based on my PowerPoints that we go over in the lecture. But having said that, I still do ask questions, a minor amount of questions coming from the textbook. So I'm not surprised that this one might have gotten a lower response, correct response, because it is the unresolved questions about early life. Which of the following is not one of those options. So they, the one that stands out is this one. Cyanobacteria, everyone agrees cyanobacteria came later in evolution. They weren't one of the first protocells. Cyanobacteria are too complicated to be the initial protocells. Here's another one. This was again assigned. I had PowerPoint slides on them, but I didn't personally go through them with you, but they were available plus the associated reading. And again, I think that might have been the reason why some people weren't picking up on this one, but the basic idea of how they were running this experiment, they were doing the seed. They were subculture each day into fresh broth. They were just plopping some aliquot into fresh broth. They just kept doing that over and over and over and over and over. They're still doing it. Here, this was from of what we talked about as well as textbook reading. This is, so all of these are true except for one statement. And so we're talking about Lynn Margulis and the end of symbiosis. So that's a true statement. C, yep, that's a true statement. D, yep, that's a true statement. So A. So in order to ID a microbe, you don't have to purify it. You don't have to grow it up and look at it. You can just grab I could just go and grab a fistful of soil and send it off and say, tell me who's in here. And they can do that. They can sequence it, just extract all the DNA and sequence it. They never even have to grow up the microbe. They can still purify. They can still identify even a mixed culture like that that's given to them. Plasmids, which of the following is true? So only one true statement here. Which one is the true statement? Thoughts? So this is, a lot of this is taken from that table I asked you, if you looked at it in PowerPoint in class, and to make sure that you do the top half of the table, pairing bacteria and eukaryotes with cell types. There's only one true statement. Anyone want to hazard a guess? No? B. That's a true statement. All the others. There are some bacteria that have linear chromosomes, but they're very, very rare. And then C and D are false as well. And this one. This one, I think, I wanted to show this one because this, I think, what I'm guessing what went wrong here, if you answered incorrectly, is you weren't reading the answers carefully enough, slowly enough, like thoughtfully enough. Really think through it here. So, which of the following statements is false with regard to material cell size and structure? So, there's only one false statement. And A is the false statement. But again, if you read it quickly, your brain would say true. But you have to pay attention to detail. And so flavonoids are not found in the cell wall. Yes, there are flavonoids, and yes, they are, good for strength, but they're not in the cell wall. And so again, just thinking through these questions, slow down. And I know I did this too when I was a student. I would, with multiple choice, I'd read the question as quickly as possible, jump down to the answers as quickly as possible so I wouldn't forget. I don't want to forget, so I better do this really quick. And you get into that habit. I don't want to forget what I have in my head here before it's, it all bubbles away from me. And so I'm encouraging you to slow down. And I actually learned the trick of, I would cover up the answers and just look at the question. And so with this, I would start thinking, okay, asking about cell size and structure. Okay, what do I know? What did we talk about? And I just start thinking a little bit about that. And then I look, because if you do that, you're triggering the right part of your brain, the right neural pathways. That way, when you look at the answer, you're not as easily thrown. Because you know what they call the multiple choice? There's one right answer typically. You know what they call all the other ones? Distractors. That's what they actually call them. And so, it's accurate. They're there to distract you so that you have to pinpoint the right answer. And so you need to slow down enough to do that. Okay, if you want to go through your exam one more carefully, more thoroughly, we can certainly do that. Okay. So, exam content. Let's go into so cell structure and function, right? Part 1. And we need to go to, so we ended up, let me reshare. So this is the point forward. We basically did exam 1 to this slide. So exam 2 is the next slide. All right. So that's the peptidoglycan details. So we looked at peptidoglycan, differences between gram-positive and gram-negative. I touched on the acid-fast. We really didn't get into any details on acid-fast. You did more of that in the lab when you did the acid-fast test. But so peptidoglycan, you can see that there's a big swath of slides here on peptidoglycan. So obviously it's important. That many slides are devoted to it. And then we got into overall structure of the gram-negative. Let me go out of this here. So lots and lots of slides on peptidoglycan details, gram-positive versus gram-negative. And then we looked at the overall cell structure of gram-negatives, the cell wall structure. A little bit more detail with that funky outer membrane. And we dissected that. And then we got into a little bit of archaea, how they can do a range of different things. But that paracrystalline is the most common, which is the picture being shown. Okay, so questions on this slide set. Peptidoglycan. Yeah. So the inner leaf of the outer is this here, where we have, it's pretty similar to a normal. The inner membrane thing is you have that lipid here. Or no, that's we have these that anchor in, so have that so that you can have it hooked to the peptidoglycan. That's basically the square here, but that's the outer, this portion of the outer leaflet of the outer membrane. So that outer leaflet is very much more complicated than the inner leaflet of the outer membrane. Other questions on cell structure and function? No, not other than, like, I'm guessing what I have what, 3 slides on it, so not much. Those I think would easily be multiple choice questions, you know, maybe 1 or 2. Anything else on? Yes. Teichoic acids, yep. Didn't we already cover that though? Oh no, here they are. So the basic idea is that when you look at gram positives, they tend to have a lot of teichoic acids in their cell wall area, interspersed in their peptidoglycan. And some are just straight integrating into the lipid layer, so that's why they call it the teichoic acids found in the gram-positive cell wall. We don't really see teichoic acids in gram-negative. And then they're negatively charged. That's about it. I mean, we didn't really get into big detail on that. So the thing that's kind of unique is that it's showing up in the gram-positive. Other questions on chapter 3? Well, this slide set. We got another slide set here too. So we also looked at part 2 of cell structure and function is looking at some like the flagella structure. So we went through that in detail in lecture. So that's something you should be definitely spending time looking at. and knowing the difference between, the paratrichus, the polar, lophotrichus. And then we did, after we kind of dissected the whole flagellar apparatus, we started looking at some other ways that they might move depending on their habitat and their strategies. Questions on any of that? Yes. Yes. So flagella structure is, usually when you say flagella, you only think about the thing that sticks out, that actually does the movement. Do bacterial flagella whip their movement? No. So in three-dimensional space, they don't whip. They're not all, the flagella is not flexible like that. It's a rigid corkscrew, and it just rotates. Right? So very, very different compared to what you've learned about most flagella so far in your life, I think. And so it's this idea that you have to, okay, you're gonna have to rotate like that. You gotta put it all together to allow it. So you have the rod, and then the hook region is angled like that, so it connects well to the hook through like filament. And the filament is just a bunch of protein that gets built in the cell. They get sent out, attached. Filament is longer and longer. But it's just a bunch of protein, and it gets set up in this rigid corkscrew like pattern. The hook connects it to the rod, and then that rod goes through the rings. And then you have the Mott proteins that are really driving the rotation. A motor, Mott motor. And so it's like the motor on a boat that's going to move the propellant. And in order to do that, though, they have to get a flow of protons. So they have to generate a proton gradient to get that flow going through the Mott protein. And if they do that successfully, it can cause the rotation of the filament. And then the only other thing on there is the cleave proteins that just allow the filament to either go clockwise or counterclockwise. So you don't have to know the letters, and you don't have to know how many rings. Just know that there's rings. You know that the rod has to. But I'm not going to get into the letters. Yes.
So the difference bacteria is flagella, we can just see this one has like the outer one.
Yeah. And so they don't even know how well they've studied gram flagella, because they've really zoomed in on the gram negatives. And so I rarely even see gram positive flagella talked about. So I'm not even sure what their apparatus is. They must have similar, I'm guessing, fewer rings. Don't have as many rings. But
So what
yeah, it's a way to, they're hollow, so it lets the rod comes through. And it's a way to stabilize the rod and stabilize the whole. It's almost, think of it like a root structure, kind of like it's rooting into the membrane, cell membrane, that's allowing for a good anchor, so to speak, I guess, for the filament to make sure that you don't have that breaking off, becoming dysfunctional. Other questions on flagella? What about any other slide contents in here? Yes. Yes.
So for
Small acid soluble dipicolinic?
yes.
Yeah.
The former complex with calcium, and then those are specific to endosperm production, and they prevent dehydration, or they do help with dehydration?
They primarily protect the DNA as it's getting dehydrated, so as the endospore starts to desiccate, you get that. Divergent assays, getting those involved to help stabilize DNA, proteins, making sure that they don't fall apart as dehydration is going on. Really amazing process, all in all. It makes it through that process. It can still get activated under the right conditions, fully functional again. Any other questions? Okay, so cell structure and function, those two slide sets. And then after that, we got into metabolism. So, and we're still talking about metabolism. So this test is going to be heavy metabolism, cell structure and metabolism. So we got into intro of metabolism. So that's where we were looking at first thinking about things that maybe you're already familiar with, the glycolysis, the TCA cycle, the electron transfer chain. What kind of questions do you have on this? Yes.
No, like baseline stuff about the glycolysis and citric acid.
Right. So let's pull up glycolysis. So remember, this is not a biochemistry class. So at least when I took biochem, I had to learn everything, all the enzymes, proper order. Do they still do that? And so that's not what we do. So I'm more interested in the key components of glycolysis and the big picture idea of what is it accomplishing and under what conditions would a microbe choose to do this. So knowing it starts with sugar, we typically talk about glucose and that you have to expend some energy. That's important. And then after you do that first step, then you get into the second phase that works it down to pyruvate. What's substantial about stage 2? What's a good thing going on there? So if I asked what's good about stage 2? Yep. And how is it generating that ATP? Very important that you understand that that substrate level is key. And then by the end of stage 2, you got pyruvate. So then what would a savvy microbe be thinking about? Okay, I've made my pyruvate. Presence of oxygen would be a good thing. Stop and ponder and assess the environment. What do I got here? Again, kind of like going into your pantry. What do I got to cook with here? I want to make the best souffle ever. Tastiest thing. I want oxygen because I'm going to get the most ATP. So that's a good place for them to just chill and assess. And then if they go, oh yeah, I got oxygen, where would they send the pyruvate, so to speak? What other pathway would they kick into? The citric acid or TCA site. But if they're looking around, they're like, I don't have any oxygen. Then fermentation is an option. There's another option we learned about, but we didn't get into it until fairly recently. So in here, looking at this, you could say, well, yeah, if there's no oxygen, you're going to go into the rest of fermentation. And that's totally fine. But do you get a ton of ATP? No. So it'll work. But we've learned about something else in later PowerPoint slides, that if they don't have oxygen, they could do something else that's better than doing this fermentation. Remember what it is? Electron transport chain. So what do we call that if oxygen isn't available? Anaerobic, right? Anaerobic metabolism is usually when they say that phrase, they're thinking about electron transport chain. It's just not oxygen at the end. Okay, so if I was this microbe, I would sit tight at pyruvate and assess oxygen. No. Nitrate, anybody? Do I have that? And if they do, that would be a good option. Stick nitrate into their electron transport, and there's one of their cell membrane apparatuses. They have an electron transport chain that doesn't include oxygen. They also have one that has oxygen at the end. And so they have all of this embedded in their cell membrane. Do bacteria have mitochondria? Once again, do bacteria have mitochondria? It's kind of a pet peeve. So don't be surprised if you get asked that question. All right, so you have this idea of the cell membrane. Hugely important because they have to put all this stuff in order to run, especially the electron transfer processes. They can be a lot of different. They don't have to be locked. Second. Usually, if they have oxygen, they'll have that one cooking, like boom, boom, boom. They'll just have it going. And then once if they depleted all their oxygen, they'll go, all right, if I have to, I'll kick into nitrate at the end. And so, but a savvy microbe will for sure run the electron transport chain section that has oxygen sitting at the end. So these are the types of things, these questions I'm asking you right now, these are kind of the big picture ideas I'm hoping you're catching, because you can take this information into any of your other classes. And if you kind of get these bigger pictures, it tends to make things fall in place a little easier as some of these pathways. So yes, they could take pyruvate and run it if they really had to, but you know, if I was smart, I would not do the rest of fermentation if I had some other terminal electron acceptor and an electron transport chain process set up for that. But sometimes they're out of luck, or they might just not have that capability. Some microbes never evolve that. They can just do fermentation. And then it can send off ATP, stay alive, and reproduce, but you have to do a lot of glucose ingestion, so to speak. You have to get a lot of that to make enough. What's another thing going on in stage 3 that's a good thing that helps keep everything going? What else is going on there? There's something else that's key that we talked about. NAD plus. NAD plus gets made here. And so NAD plus NADH is not, there's not a lot of that in cells, including bacteria. And so they constantly have to find ways to keep recycling it so that they push these reactions. So you're seeing in stage 2 that NADH gets made, and that can help stage 3 go to completion, and then that NAD plus can come here and stage 2 go to completion. And just keep doing that all day long if they're running, if they get fed a lot of glucose. So those big vats, you know, when you go to a beer brewery thing, you do the tours, that's going on big time. And they feed a lot of starch or glucose to those yeast in order to keep this going. What other questions on this slide set? So let's go to this one. Okay, so again, for this, I'm not asking you to know every step of the way. I'm not asking for all the enzymes, any enzymes really. But thinking about the TCA cycle, there's some key things that are really good about this cycle that really help the microbe doing it. So it's important that you know what kicks it off. In our case, let's talk about pyruvate. Okay, there's other things I can feed in, but let's zoom in on pyruvate. So that pyruvate's coming from glycolysis. And this is where the microbe's like, hey, I got oxygen. And that is awesome. Well, I'm going to send my pyruvate to TCA because the TCA product is going to help me take advantage of oxygen. So what are some of the products here, just in general, that we should say, yeah, that's a good thing for the microbe? Well, the CO2 is really good. If you can demonstrate that that pyruvate, which is an organic molecule, got completely broken down to CO2, if you do the energetics of that, that's really good. It's going to set them up for good ATP production. So that's definitely good. And fermentation doesn't do that. You don't see the starting substrate getting broken all the way down to CO2. Some CO2 might get it, but it's not a complete breakdown. Okay, what else do people? ATP or DTP? Good, for sure. That's a really good thing for the cycle to be able to make. What else? Yep, FADH2, NADH. So why should we care about it? What are those called? There's several different terms for what NADH and FADH2 are called. Reducing agents. What do they do? Like, what's their job? Donate electrons. So when they're in the NADH state, the FADH2 state, they're fully reduced. That means they got electrons that they can then move to another pathway and donate off. And so their job is to donate off electrons into some pathway. And so by the time the TCA is done, you've broken down pyruvate, that's great energetically. When you do the energetic good. You get reducing molecules, those are really good. You get GTP or ATP, those, that's good. So now let's think how that links. So what are they going to do now that E. coli is like, yay, I have oxygen. I'm going to go put my pyruvate into the TCA, run this, and then I'm going to take my NADH and my FADH2 and put it where? Yeah, put it into the electron transport chain, because that is where they really make the souffle. Okay, they really get it to the point where they maximize their ATP. So that's being shown here. And so the NADH and the FADH2 are feeding in. And be aware that, you know, microbes have different flavors of electron transport chain. This is showing one type. There's the NADH, there's the FADH2. Okay, so those are coming from the TCA. And the microbe says, all right, you know, I made some reducing agents, go do your job. So their job is to hook into the electron transport chain and start donating off electrons. And so you have these different donors and acceptors in the membrane, passing those electrons, kind of like a hot potato. And you have some terminal electron acceptor for the microbe because they're like, ooh, I have oxygen. This is the one they chose, the one that has oxygen as the terminal. And so as the electrons are flowing, you see them going, you know, initially from NADH, FADH2, all that's feeding in. And then finally oxygen gets the electrons and gets reduced to water. Is that when the microbe goes, yay? No, that's not the souffle. It's just water. And although water is important, that's not their goal. All right, so they evolved this little game to play to take advantage of oxygen, because as they do the electron transport chain with oxygen, they're getting the best ATP production. But how are they setting up ATP production? What, along with electron flow, what's linked to electron flow? Proton pumping, which leads to a proton gradient. So electron flow, proton pumping, electron flow, proton. And so really the game is I'm going to create these donors and acceptors in such a way that energetically it creates situations where I can use a little bit of energy to pump protons. And that's all they're doing. They're just playing a game to create that little bit of extra then they get that proton gradient, which isn't super obvious here with the way the artist drew it, but you should see a lot of protons building up here. So as long as they keep this electron transport chain going, they get that proton gradient. And once that's established, you got to finish the thought. You can't just leave it at a proton gradient, especially if I ask an essay question on it. You got to finish the thought. So what? Proton gradient. You got to pretend like I don't know what that means. Okay? So you have to link it that, oh yeah, when they form that proton gradient, that can be really good. Because then as the protons go through that ACP synthase, it allows for ADP and inorganic phosphate to react. So then you get ATP. So we said that glycolysis is substrate level phosphorylation. What is the electron transport chain? Oxidative phosphorylation. That's another huge. So substrate is a huge concept, substrate level phosphorylation, oxidative phosphorylation. But it's asking, how is ADP getting phosphorylated, right? Is it substrate or oxidative? What does oxidative really mean though? In a microbe world, what does oxidative mean? Stripping electrons, right? Does not necessarily mean oxygen is involved. Now, in this case, yeah, we've just talked about oxygen. But when you hear oxidative phosphorylation in this class, don't assume it's oxygen at the end. And I know that's a hard flip to make when you're so used to thinking about how animals, humans, we do oxidative phosphorylation in our mitochondria, but it's different. Microbes can do oxidative phosphorylation with nitrate sitting at the end. Still oxidative phosphorylation. Okay. Other questions? What about the next slide set on so then we got into fermentation, a little bit more detail, and anaerobic metabolism. Is this the right slide set? Questions on anaerobic? What about the oh, yeah.
Denitrification and genesis,
Where
that's just ways of
Good question though. So denitrification, let's look a little bit carefully at the figure here that they're showing with it. And if you just see the reaction pathway, sometimes I have to really slow down and think. But if I see it like this, it's much more obvious what's going on. And so with denitrification, you look at how they've drawn this out. This is denitrification because it's showing that you have the reduction going all the way to dinitrate. So if you take nitrate and keep flowing it through, reducing, and you get all the way down to dinitrogen. That's denitrification. Going from nitrate to dinitrate. But let's look at how the artist drew this. What kind of overall process is going on here? What are the black lines implicating? Yeah, electron transport chain. You got players in the game. You have these different complexes in the membrane. And then you see electrons going from each one. And so if you just think, kind of tease that apart, it's like, okay. And again, fermentation tends to take place in the cytoplasm, whereas electron transport chains tends to happen in a membrane. For us, it's our mitochondria. But for bacteria, that's so if you look at it, go, oh, okay, yeah, they're drawing electron transport with these different donors and acceptors. And they're showing proton pumping across the membrane. Boom, that's an electron transport chain rather than fermentation. Other questions, we did anaerobic, we looked at chemolithotrophy. What's the other, what the other slide set, we looked at fermentation more deeply. Where is that? I must not have that pulled up yet. That one? No. Yeah. Okay, so we also looked at this one with fermentation, the redox tower idea as well, to get you thinking about what helps, which reaction pathways give you better free energy. Questions on the tower or anything in here? Redox, we talked quite a bit about redox. Yeah. Now, so you know these iClicker questions we did, like that would be, I might ask something like that, where you have to think through, I would give you enough information where you could tease apart who is donating, who's accepting, but not to actually memorize the tower. That's something you can easily look up in multiple different resources. Yes. Mhm. So with the iClicker, we were looking at, so go back to this real quick. So we were looking at this idea of whatever's to the left of the slash is the oxidized form, whatever's to the right of the slash is the reduced form. So you have to know that rule. And then the farther the electron falls down this tower, the more free energy they will get. And so this slide is showing that, where they're comparing free energy of 3 different reactions. So that's an important concept. Farther the electron flow falls, the more free energy it will get from that. So we started to look at a couple of options in the iClicker here, where we were looking at if you have a pair, CO2/acetate pair, and then the Fe3+/Fe2+ pair, and then the reduction potential being provided, one is a negative and one is a positive. And so if it's a negative reduction potential, that means one of those players is good to go for donating. They can donate on, but you have to know which one, CO2 or acid. So that's where you have to know that by convention, what's to the left of the slash, what's to the right of the slash. So which one is capable of donating? The oxidized one or the reduced one? If it's reduced, that means it has enough electrons where it can safely donate. And so acetate is to the right of the slash, the reduced to the right of the slash of the acetate that can actually donate. So if it's first of all, this particular slide is just saying, I just want you to key in on what is the reduction to answer this question. That's what you're looking at. Like, okay, I need to figure out, okay, reduction potential is negative, acetate is positive. So by the rules of the game, you look at the negative one. One of those, CO2 is going to be capable of donating. So A is the correct answer because it has a reduction. The reason it's correct is it's got a negative reduction potential. Then I said, okay, now that we got that figured out, which one of them is going to actually be capable of donating? By convention, the one to the right of the slash is reduced. Reduced means it can give off electrons. That means acid is the correct answer. Now, the way this is set up, I'm applying that in this case now acetate is gonna donate electrons to either Fe3 plus or Fe2. So then it's like, okay, if acetate donates off electrons, then it's gotta donate to something that's oxidized and needs electrons. And so then thinking about that, then you have to zoom into, okay, which of these Fe3 plus or Fe2 plus is ready to take on an electron, which means it's oxidized. Like, give me an electron, I need an electron. So which one of those? Fe3 plus is going to be oxidized. The left of the slide. So Fe3 plus is the one to get that. So it'd be acetate. Is going to donate plus Fe3 plus will accept. CO2 will be a product and Fe2 plus will be a product. And if I ask you something like this, I'm not going to ask you to balance it. Do the stoichiometry. All right, you need to know where the cutoff is. I'd like to tell you that now, okay? So we're covering the, let me get out of this. Is this the one that's, yeah. So it will go through what halobacterium is doing. So technically not photosynthesis, we're stopping here. Okay, I'm going to start photosynthesis, true photosynthesis on Wednesday, but that will not be on the exam. That is not enough time to figure out photosynthesis. Okay. So it's through to the halobacterium. So Wednesday, I will kick into photosynthesis. I'll have a little bit of time. I can answer any lingering questions. Okay. But I really, really want you all to do well on this next exam and feel like, you know, it's starting to fall into place. But that takes time. Remember, you're building new neural pathways. You can't do that overnight, especially with some of this abstract stuff. You have to get at it more than once, for sure. Okay. All right. So I'll see you on Wednesday. Yeah, I can do that. Do the tips and tricks type thing.